What's up guys, Omio here once again. I'm sure you guys have already heard the word antibiotics somewhere along the road and have some basic information regarding this topic. But in this presentation, we're going to take a closer look at the mechanism, classification and nature of antibiotics. Antibiotics were first discovered by Sir Alexander Fleming in 1928, but we already know that. But his discovery was actually an accident, but it was one of the greatest accidents in 20th century. At that time, the antibiotics were used to treat infections caused by any microorganisms, not just bacteria. But in modern times, the definition of antibiotics has changed. We use now antibiotics to treat bacterial infections like pneumonia, tuberculosis, bacterial meningitis, and many more. So antibiotics are used to treat bacterial infections. So that means they are not effective against viruses or other microorganisms. So how do antibiotics work? Like, how do they do what they do? They either stop the reproduction of bacteria or kill the bacteria. Antibiotics that stop or interfere in the reproduction of bacteria are called bacteriostatic, and those which kill the bacteria are named as bactericidal. Keeping these terms in mind, let's take a look at the classification of antibiotics. There are 6 main classes of antibiotics based on their activity mechanism. The first class of antibiotics, which are also the most common ones and the most known ones, are the penicillins. Penicillins are bactericidal. That means that they kill the bacteria, Terminator type shit. You have to keep in mind that they are only effective against gram-positive bacteria. That means that they kill only the gram-positive bacteria. Penicillins have a beta-lactam ring, which looks like this red marked ring in the center of this penicillin molecule. All the different types of penicillins have this core ring structure in their center. And this ring structure is actually the beta-lactam ring, and that's why they're also called beta-lactam antibiotics. Penicillins kill the bacteria by inhibiting the biosynthesis of cell wall. But how do they do that? We all know that bacterial cell wall is made up of murine or peptidoglycan monomers. So the beta-lactam ring inhibits the cell wall synthesis by inhibiting the peptidoglycan synthesis. They do this by inhibiting the activity of an enzyme called DD transpeptidase, which facilitates the cross-linking or transpeptidation of peptidoglycan of bacterial cell wall. The transpeptidation of bacterial cell wall is the reason why the cell wall of bacteria is so rigid and it can uphold the osmotic pressure within the cell. Normally, the bacterial cell wall constantly needs to be transpeptidated, or in other words, renewed. When the transpeptidation of cell wall is blocked, the newly formed cell wall isn't strong enough to hold the cell components intact. Because of the osmotic pressure within the cell membrane is ruptured, and there is no functional or rigid cell wall there to hold the osmotic pressure within the cell. This causes the genetic material, cytosol, along with other cell components to spill out of the cell, leading the bacteria to cell death. This molecule here is a carbonyl donor during peptidoglycan synthesis. It is a vital component in cell wall synthesis and is the substrate to the enzyme DD transpeptidase. And this right here is a beta-lactam ring. As you can see, they both look very similar. And because of the similarity, the beta-lactam ring binds to the DD transpeptidase and inhibits its catalysis. But sometimes the bacteria find a way to survive the penicillin. In other words, they become strong against the beta-lactam antibiotics. They start producing an enzyme called beta-lactamase, which makes them resistant against beta-lactam antibiotics. So what this enzyme does is, it hydrolyzes the beta-lactam ring. When the beta-lactam ring is hydrolyzed, the enzyme DD transpeptidase isn't inhibited anymore, which means it can synthesize peptidoglycan and form a rigid bacterial cell wall which is necessary for its survival. Here are some common examples of penicillins like ampicillin, penicillin G, cephalosporins, which are actually a class of penicillins because their working mechanism is the same as the penicillins and they are beta-lactam derivatives. That's why they're considered a subclass of penicillins. Sometimes, however, they might be considered a separate class of antibiotics like penicillins, but mostly they are considered as a subclass of penicillins, like their derivatives, which is also the case. Other examples of penicillins are amoxicillin, oxacillin, and many more. Microlides are the next category of antibiotics we're gonna look at. They're effective on gram-positive as well as gram-negative bacteria. They have a wider antibacterial spectrum than the penicillin. They can also be used as penicillin substitutes, but their working mechanism is a little bit different than the penicillin. They inhibit the protein biosynthesis by blocking an enzyme called peptide transferase. Peptide transferase is the primary enzyme in the ribosomes, which forms peptide bonds between two adjacent amino acids using tRNA during translation. The macrolides block irreversibly the ribosomal subunit where the enzyme is supposed to catalyze the binding of two amino acids. And by the way, the macrolide activity is bacteriostatic. Here are some examples of macrolides, the most common one being the erythromycin. The next category of antibiotics are fluoroquinolones. Antibiotics from this category kill the bacteria and are effective against both gram-positive as well as gram-negative bacteria. They do this by interrupting the unwinding and replication of bacterial DNA. This is how fluoroquinolones look like. But how do they stop the replication of DNA? They interfere in the DNA replication by interrupting the enzyme activity of topoisomerase, an enzyme class that unwinds the DNA, and ligase, which joins the two DNA strands together. So normally the topoisomerase unwinds the DNA, but when this enzyme is inhibited, the DNA can't unwind, which means that the replication of DNA can't take place because the DNA needs to be unwinded first. And also transcription is not possible in this case because the DNA needs to be single-stranded in order for a messenger RNA to attach and transcript the DNA strand. And the inhibition of ligase results in dysfunctioning of the DNA, which means that the DNA can't control the cell and the necessary proteins cannot be produced by the cell, which eventually leads to cell death. Some common examples of fluoroquinolones are ciprofloxacin, levofloxacin, and more. Tetracyclines have 4 hydrocarbon rings. That's why they are called tetracyclines, because they have 4 hydrocarbon rings. Different types of tetracyclines can have different functional groups attached to them. This is what a tetracycline looks like. They have a very wide antimicrobial spectrum, including some cyanobacteria and some unicellular eukaryotes. They're effective against both gram-positive as well as gram-negative bacteria, and they are bacteriostatic. They interfere with the protein synthesis of bacteria. You might think, how do they do that now? They stop the initiation of translation by binding to the 30S subunit. They inhibit the binding of amino-acyl tRNA to messenger RNA, which is necessary for initiation of translation. Amino-acyl tRNA is a transfer RNA complex that delivers single amino acids to the ribosomes for polypeptide formation. Some common examples of tetracyclines are tetracycline, oxytetracycline, and many more. The next subclass that we're going to look at are aminoglycosides. Mostly bactericidal, but sometimes they can be bacteriostatic depending on the species of bacteria and concentration of these antibiotics. They're effective against most gram-negative bacteria. Aminoglycosides are protein synthesis inhibitors. They inhibit the protein synthesis of membrane proteins by disrupting the peptide elongation at 30S ribosomal subunit. The disturbance in the peptide elongation leads to inaccurate translation, which results in dysfunctional peptide chain, which causes the ribosomes to stop the translation before the peptide chain is fully functional. The cell membrane, when incorporated with these dysfunctional proteins, face changes in its permeability, which means that the cell cannot control the in and out diffusion of substances or molecules in and out of the cell, and a gradient inside the cell is not possible, which may eventually lead to cell death. One thing to remember here is that aminoglycoside activity produces dysfunctional proteins, but translation does occur. In contrary to tetracyclines, which stop the translation initiation, which means that the translation does not take place in that case. That's the main difference between the two. Here are some common examples of aminoglycosides. The subclass of streptomycins are considered to be aminoglycosides, and some common examples of aminoglycosides are erythromycin, gentamicin, and etc. The complete class of sulfonamides are synthetic, but the other subclasses of amino acids are extracted from living organisms and then produced synthetically, but this class is completely synthetic. The antibiotics in this class act bacteriostatic and are effective against both gram-positive and as well as gram-negative bacteria. Most sulfonamides inhibit the folate synthesis in the cell. Folate is also known as vitamin B9. Vitamin B9 or folate is actually very necessary for dNTP or nucleotide triphosphate synthesis. These are the nucleotides that are needed for DNA or RNA synthesis. So no folate synthesis, leads to no DNA or RNA synthesis, which means that there is no DNA replication and there is no protein synthesis because the RNA cannot be synthesized, and hence no transcription and translation. Here are some examples of sulfonamides. Sometimes the bacteria become resistant to the antibiotics, so they develop enzymes against these. Different species of bacteria develop different types of antibiotic resistant enzymes. Some bacteria develop hydrolysis enzymes which hydrolyze the antibiotics, like penicillin resistant bacteria develop beta-lactamase, which dissolves or hydrolyzes the beta-lactam ring and hence making it ineffective. Some bacteria produce enzymes that inhibit the antibiotic activity and some produce deactivators. In some cases though, bacteria produce substances that protect the receptors from antibiotics, which means that they cover the substrate to which the antibiotics are supposed to bind. So basically the type of resistance that the bacteria develop depend on the species and circumstances that the particular bacterial species faces. Different classes of antibiotics have different side effects. The most common side effects of all antibiotics include vomiting, nausea, diarrhea, bloating or indigestion, abdominal pain, loss of appetite, and itching. For specific side effects, just read the labels. Thank you guys very much for watching. If you guys still have some tips for me how to make my video quality better and what you want me to improve the first or the most, please let me know in the comments, and I'll see you guys in the next one.